In this lecture, we're going to talk about another type of division called meiosis. Most animals undergo a specialized division called meiosis to produce gametes, sperm or egg, to allow for sexual reproduction. This is a picture of a sea urchin egg with tons of different sperm ultimately trying to bind to the egg and enter so that fertilization can occur. Sperm and egg are produced by specialized cells called germline cells. Germline cells will undergo meiosis to produce sperm or eggs. Sperm or eggs are called haploid cells, and haploid cells refer to the fact that they have only one of each type of chromosome. So humans have 23 pairs of chromosomes. In their haploid cells, their eggs or sperm, we have only 23 chromosomes, one of each type. However, after fertilization between sperm and egg, now we end up with two of each type of copy, one copy donated from the egg, one copy donated from the sperm. So that's why we end up with 23 pairs, and ultimately that results in what's called a diploid zygote. Diploid referring to the fact that now we have two of each type of chromosome. So in humans, that would be two of each of the type of chromosomes to 23. Then, once this zygote is produced, cells will undergo mitosis to produce identical cells that will ultimately result in a ball of blastula cells that will then grow and develop into a specific organism. However, during this division process, we'll also set aside some germline cells that ultimately will allow that organism, when they reach adult form, to produce both sperm and egg from those germline cells. The cells that are not cells that will undergo meiosis, meaning cells that will ultimately always divide by mitosis, are called somatic cells. This figure on the right is showing the number of chromosomes and the reduction in chromosomes during this event. So ultimately, mother and father will produce sperm or eggs by meiosis, resulting in haploid cells. So you can see the reduction in chromosomes. Originally, the mother and the father both had two, and in this egg and sperm, now they each have only one. And then upon fertilization, union of these two gametes, you once again restore the chromosome number of two, one of each type, two of each type, of the one, and then that cell will then undergo mitosis to produce a multicellular organism. So when we look at formation of a zygote from two haploid cells, the zygote now being diploid, that zygote will then undergo multiple cell divisions, some of which will result in a set of cells that end up being set aside called germline cells. These germline cells will end up in the sex organs, the testes or the ovaries, and they'll ultimately divide by meiosis to produce new haploid sperm or eggs or gametes. The rest of the cells will continue to divide by mitosis to produce the somatic cells, the cells that ultimately will produce all the other parts of the body. To emphasize this even further, we can see in adults, male and female, the germline cells residing in the sex organs, which will lead to production of haploid egg and sperms due to meiosis in those germline cells. Ultimately, fertilization will lead to production of a zygote or a fertilized egg, which is now diploid, meaning two of each type, which is designated 2N because two of each type of chromosomes. Then that cell will then further divide by mitosis to produce all the other cell types of that organism and set aside some of those cells for germline cells. Mitosis will continue to produce an adult form of that organism. So just to remind you, these sex cells are haploid, half the amount of DNA as the original organism. However, union between two sex cells, such as a sperm and egg, will once again result in a diploid zygote, an organism that now, a fertilized cell that now has two of each type of chromosomes, which means it's 2N. So what is this meiosis process? And how does it lead to ultimately reduction in chromosome number to produce a haploid gamete? And ultimately, meiosis has two divisions. The first division is called meiosis I, which is shown here, a cell that prepares for division, duplicates its chromosomes, moves into M phase, will divide into 2 cells. Those 2 cells will then enter into a second division, and the second division will not have an additional DNA replication phase, so instead those chromosomes will divide once again, and we're left with a half the amount of chromosomes as the original cell. In this case, it's showing 1 chromosome in each gamete when the original cell had 2. These 2 cycles are also shown in this slide as well, where ultimately a cell will prepare for meiosis, duplicate its chromosomes, and then ultimately enter into meiosis I. The DNA will divide between 2 cells, and then the DNA will divide once again in meiosis II to produce 4 cells, now with half the amount of genetic material as the original cell. These are considered haploid and haploid gametes. Now, you'll also notice that there seems to be some mixing of coloration, which is supposed to represent that during this meiosis process, we have mixing of actual homologous chromosome or parts of chromosomes, which you're going to learn happens in meiosis I, in prophase I, and it's due to crossing over between homologous chromosomes. So as a comparison with mitosis and meiosis, remember we just learned mitosis, and ultimately mitosis is going to start with a diploid cell and then divide by mitosis and produce 2 daughter cells that were identical to the original, so essentially they're clones. And these cells at the end are considered diploid because they have 2 of each type of chromosomes. In meiosis, instead, there's 2 separate cell division processes, which instead of getting 2 cells results in 4, and these cells, now called gametes, are haploid in nature because they have only 1 of each type of chromosome, which we designate N. Now, why is it that something like sexual reproduction has evolved when other organisms engage in what's called asexual reproduction? Asexual reproduction, if you remember, bacteria, other types of organisms as well can engage in asexual reproduction, but it only results in identical clones or copies of the organism itself. Meiosis and the ability to ultimately produce gametes for sexual reproduction allows for genetic diversity. And we're going to talk about all the different ways in which meiosis generates genetic diversity. This seems to be advantageous because many animals, or most animals, and plants, etc., engage in meiosis to allow for sexual reproduction. And there's some reasons, one of which is genetic diversity, which allows potentially for organisms to have a greater potential survival in a changing environment. Second of all, sexual reproduction also helps get rid of deleterious genes that might pop up due to mutational changes, etc., and ultimately that can speed up its removal. If a deleterious gene popped up in an organism that only reproduced by asexual reproduction, then that particular change in gene, which would then be passed on to new organisms that were identical in nature, and ultimately that could lead to their destruction of that particular population. So ultimately, sexual reproduction has different advantages, and which is why it has persisted over time. So this shows you the differences between mitosis and meiosis. Remember, once again, mitosis results in 2 cells that are identical to the original cell, whereas meiosis, we're going to end up with 4 cells due to 2 divisions, and ultimately these cells are going to be haploid instead of diploid due to the fact that we have 2 divisions and only 1 DNA duplication phase, which leads to reduction of chromosomes to 1 of each type of chromosome, so half the amount of the original cell. And you'll also see each of these gametes end up being genetically distinct, which we're going to talk about how that actually happens as well. So not only do we have half the number of chromosomes, 4 cells instead of 2 in comparison to mitosis, but we also have genetically distinct gametes from one another. So let's start the cell division process. In germline cells, they would essentially go through G1, the chromosomes would become duplicated in S, and the cells would enter G2, and then the cells enter M phase, but this time they're actually going to go through meiosis. A G2 cell will then enter into meiosis I, and the first phase of meiosis I is prophase I. You'll notice there are early, middle, and late parts to prophase I, depending on how far into prophase the cell actually is. The key events that happen in prophase and mitosis are some of the same key events that are going to happen in prophase I of meiosis. The mitotic spindle is going to form, and chromosomes are going to condense. One big difference, however, is you'll see that homologous chromosomes synapse. And homologous chromosomes are chromosomes that are in similar size, shape, and contain similar genes. These are chromosomes you receive, one from your mom and one from your dad. These types of chromosomes, homologous chromosomes, will pair up, and this will allow for what's called crossing over. And this allows for exchange of information or mixing of your chromosomes that you have before you divide them and pass them off to your gametes. Prophase I will then lead to prometaphase, which isn't shown here. So the nuclear envelope will break down and chromosomes will start to attach to these synapsed chromosomes. Then the synapsed chromosomes, these homolog pairs, will then align at the metaphase plate in metaphase I, which you can see here. This metaphase cell will then have chromosomes be divided, and homologous chromosomes will move to opposite poles. After anaphase I, then we have telophase, where the nuclear envelope will start to reform around these separated homologous chromosomes, and we will then lead into cytokinesis, which will end up with our first two haploid cells. These two cells will then enter into meiosis II, and thus starting prophase II. The mitotic spindle will reform, and chromosomes will be condensed, and then move on to prometaphase, and then into metaphase II. And you'll notice in this particular scenario, these now chromosomes, not homologous pair chromosomes, but duplicated chromosomes, now align on the plate. And this looks very similar to metaphase of mitosis. However, due to crossing over, you'll notice there are some changes to the actual chromosomes as far as mixing of DNA. These two cells, now in metaphase, will then enter into anaphase, where sister chromatids will separate to opposite poles in both cells, and telophase, where the nuclear envelope forms around the separated masses of DNA, and then followed by cytokinesis, where we have 4 gametes produced. Each of these gametes will now have half the chromosome number as the original cell, which is why we call them haploid. Now, the scenario that we just went through for meiosis I through meiosis II is exactly what happens in sperm. We start in the testes, and ultimately one cell will lead to the production of 4 haploid gametes. However, oogenesis, which is oocyte formation or egg formation in females, is a little bit different. You'll start with a cell that will undergo meiosis I, and ultimately meiosis I will produce 2 cells. Now these 2 cells, you'll notice, are different there's a difference in size. One really large cell and one really small. The really small cell is called the polar body, and the second cell is the secondary, what's called oocyte. Now this secondary oocyte will then divide again by meiosis II to produce 2 new cells, one of which is the polar body, and the second of which is the final mature egg. So at the end of oogenesis, egg production, you only end up with 1 viable cell rather than 4. But the cell has still gone through 2 division processes to result in reduction of chromosome number. So ultimately, this cell will still be haploid in number. The polar bodies that are produced from those 2 events will get absorbed and destroyed by the body. and we're left with just one viable egg. So unlike sperm, who end up with 4 viable haploid cells at the end of meiosis, oogenesis results in one viable, very large egg. And this large egg contains all the nutrients needed once fertilization occurs so that ultimately cell divisions can occur prior to implantation into the womb and development actually of a placenta for delivery of nutrients to the fertilized egg. Now let's look at crossing over, the event of crossing over, which happens in prophase I of meiosis. So remember, you get this pairing of these homologous chromosomes that are similar length and size and have similar genetic information, with the exception of the X and Y, the sex chromosomes. So in this particular scenario, you're seeing that red happens to be the chromosome inherited from the mom, the blue happens to be a chromosome from father, which we call maternal and paternal. The homologous chromosomes will synapse, and when they form the synapsis structure, it is called a bivalent structure. You can see the duplicated chromosomes for the maternal chromosome and also the duplicated chromosomes for the paternal. What will happen is specific segments of the maternal and the paternal chromosomes will actually cross over, and they will ultimately be switched, which will allow for a mixing of DNA of your maternal and your paternal chromosomes. In this case, you can see that there's not only one crossover event, but 2. These points of crossover are called chiasma, and if there's multiple chiasma, they're called chiasmata. Most chromosomes, when we think about in human egg or sperm production, have between 2 to 3 chiasmata per homologous pair. So this is something that's obviously happening in prophase I, and not only does crossing over allow for mixing of chromosomes between paternal and maternal homologs, but it also helps keep the chromosomes together until they're further separated in the end of meiosis I. This is showing you a little bit more detail of these potential crossover events. In this particular bivalent structure, you'll notice there's actually 3 chiasmata, and an electron micrograph that represents a chromosome that was drawn in cartoon here is shown to the right, highlighting obviously these important crossover points. Now this particular diagram is actually showing you labeled paired homologous chromosomes, and the yellow dot is a protein that recognizes crossover points, and you'll notice these chromosomes have at least 2 to 3 crossover events. Ultimately, crossing over results in exchange between homologous non-sister chromatids, and so ultimately you're going to see the switch here. And then once they're separated, they now have actually switched genetic material. This process of switching genetic material is called recombination. This shows you a little bit further on in the process. In the beginning of metaphase I, they're all aligned at the plate. You can see that these crossover events actually help hold these homologous chromosomes together. And then before ultimately transition into anaphase I, the arms of the sister chromatids will be unglued and essentially allow for separation of these homologous pairs. And then when we get into the second division phase, metaphase II, now we're left with sister chromatids that will then separate to opposite poles in anaphase II. Now this particular diagram is showing you in more detail all the events of meiosis. But you'll notice something. All of these red arrows here happen to be actually prophase I. Prophase I, depending on how far progressed it is, has specific names. Not any of these names that you need to know, but ultimately there's specific names that represent how condensed the chromosomes are and how far along they are in the synapsis process, which you can see from these particular cell types here. Now, prophase I is prolonged, so it's extended compared to mitosis in meiosis, and that's to allow for this elaborate crossover event to occur. And then, obviously, proceeding forward into the rest of the phases of both meiosis I and also meiosis II are about the same time frame as it would be in mitosis for that particular cell type. This is showing you once again specific cells in those specific phases. You can see leptotene, zygotene. These are all early to more late phases of prophase. And then you can see the cells moving into metaphase I, etc. This happens to be cells that are young pollen grain cells that were caught at specific phases of meiosis so that you can actually see real cells and what they might look like at these particular phases. However, all that you can see of these particular cells are stained chromosomes. Other parts of the cell have not been stained to allow you to identify them. So it's important to note that there's some important factors that help contribute to genetic diversity of offspring due to the process of sexual reproduction. Obviously, the first one we've discussed happens to do with recombination or crossing over. And this is something that happens during gamete formation. So each gamete, when it's produced, ultimately will have crossover events. And so this leads to genetically diverse gametes before we even get fertilization. However, there's another process called independent assortment that also contributes to gamete diversity in gametes. Independent assortment has to do with the random alignment of homologous pairs during metaphase I. This leads to a potential mixing of chromosomes into the final gametes, which we're going to see in a second. And finally, obviously, one of the last events of the whole process of sexual reproduction, which is fertilization, obviously, genetic information from two different parents combine together to create a final offspring, making that offspring genetically diverse from the parents themselves. That obviously contributes to the genetic diversity of the offspring as well. So independent assortment, how it applies is you have these synapsed homologous chromosomes that ultimately will end up lining up at the metaphase plate in metaphase I, which you can see from this part here. You'll notice that maternal is written in red, paternal is written in blue. And you can see that there's different possibilities in which these synapsed homologous chromosomes would actually line up on the plate. And this is a random process. It just depends on what microtubules have grabbed them from what pole first, and which will end up being ultimately how the alignment is decided. So essentially these chromosomes could have lined up this way, or they could have lined up this way. Now let's look at the outcome. If they lined up this way, and then the cells undergo another second division, you'll notice this is the outcome for the actual gametes. You'll notice two of the gametes actually have paternal chromosomes in it, and the other two actually have maternal. They still have one of each type. However, they have obviously one of your parents, and the other ones have the other of your parents. However, what happens if we mix them in this fashion? You'll see that the cell will divide and then divide again, and now we produce gametes that have a mix, one chromosome from maternal and one chromosome from paternal. They still have each one of each type, which makes them haploid. However, you'll see that they'll have a different combination than what was actually seen in the other side. This is the contribution of independent assortment. Now you're just seeing two pairs synapsed and what the possible outcome could be. Now imagine something like us, and we have 23 synapsed pairs, and you can imagine the combinations of potential chromosomes in your gametes would be vast. So what does this mean in reference to genes? Remember, these chromosomes carry certain genes, and these genes code for proteins that help contribute to the outcome of a trait: eye color, hair color, skin color, height. When we look at a comparison just thinking about genes that might be carried on the chromosomes, you can imagine what kind of difference that would be as far as gametes. If chromosomes lined up this way and the genes were A, B, and C, the large letters were on one side, A, B, and C, the recessive versions of the genes, were on the other, when they actually divided later, you would end up with more recessive genes in some gametes and more dominant in the other. But if these were more mixed up, you can see recessive and dominant genes would be mixed, and so forth. So you can see these different combinations that would occur as far as gene pools in these gametes, depending on the actual alignment of these pairs of homologous chromosomes. Now finally, the third important event, which is something that doesn't happen during gamete formation, but after. So the first two events, when we think about crossing over and also independent assortment, are both events that happen during gamete formation. So it leads to genetic diversity of just the gametes themselves. But obviously the final contributor to genetic diversity for a final offspring is the fact that you're joining these gametes from separate genetically distinct parents. And ultimately, that allows for genetically diverse offspring. Now, this is just a comparison to remind you some of the key differences between mitosis and meiosis. Remember, mitosis is going to occur in organisms in places where you need to produce cloned or identical cells. In fact, most of the cells in your body, when they need to replenish and replace tissue or grow and develop, are obviously going to divide by mitosis to produce cloned cells. However, meiosis is a process that's distinct for production of gametes, and this is localized in animals to very distinct locations. Germline cells in the sex organs will divide by meiosis, which involves two separate division events to ultimately produce cells that had half the amount of chromosomes, meaning haploid in nature, and that are genetically distinct. This will allow for ultimately generation of offspring that are genetically distinct from the parents and also from siblings of those same parents.